Hello friends, welcome to Science Made Easy and our today's topic is electron transport chain. Electron transport chain is a process which comes after the Krebs cycle and it is a series of compounds where it makes use of the electron from the electron carrier to develop a chemical gradient. It is a process that converts the redox energy available from the oxidation of NADH and FADH2 into proton motive force which is used to synthesize ATP through the ATP synthase complex, and this process is known as the oxidative phosphorylation. So, electron transport chain, it is a series of protein complexes and other molecule which transfers electrons from the electron donors to the electron acceptor via the redox reaction and couples this electron transfer with the transfer of proton across a membrane. Many enzymes in electron transport chain are embedded within the membrane, and here the energy from the redox reaction creates an electrochemical proton gradient which drives the synthesis of ATP molecule. And the main thing is that here, the final electron acceptor in case of the aerobic respiration is molecular oxygen, and in case of the anaerobic oxygen is other than oxygen, maybe for example sulfate molecule. So the electrons are transferred from electron donor to electron acceptor, leading to the production of ATP. And compared to any other step of the cellular respiration, here at the electron transport chain, the largest amount of ATP is produced in this phase. So here it is, and specifically, and one more important thing about the electron transport chain is that it is the only phase in the glucose metabolism that makes use of the atmospheric oxygen. And here the electrons are passed from one component to the another until the end of the chain, and electron reduce the molecular oxygen into the water molecule. So electron transport chain includes a number of complexes, namely complex 1, complex 2, complex 3, and complex 4. Here, the word complex defines a structure that comprises a weak protein, molecule, or atom that is weakly connected to a protein. And for example, the complex 1, it is it comprises enzymes consisting of iron, sulfur, and the flavin mononucleotide. And here two electrons are carried out to the first complex aboard NADH. Complex 1 specifically use the NADH molecule, which has been formed during the Krebs cycle. Then the complex 2, which is also known as the succinate Q reductase or succinate dehydrogenase. Here, this complex 2 only accept the FADH2 molecule, and which has not been passed through complex 1 and directly passed through the complex 2. So the second and the first complexes are connected to a third complex through a compound ubiquinone Q, and the Q molecule is soluble in water and moves freely in the hydrophobic core of the membrane. Now, the complex C, which is also known as the cytochrome C reductase, and it is comprised of the FeS protein, cytochrome B, and cytochrome C proteins, and cytochrome protein consists of the heme group, and complex 3 is responsible for pumping proton across the membrane. Now comes the complex 4, which is also known as the oxidase, cytochrome oxidase. It is comprised of cytochrome C, A, and A3, and there are two heme groups where each of them is present in the cytochrome A and A3. It is responsible for holding oxygen molecule between copper and iron until the oxygen content is reduced completely. Okay, so here in this particular image, we can see the different regions showing the intermembrane space, the second region, the inner mitochondrial membrane, and the matrix region. Here, as we can see that this is the complex 1, this is complex 3rd, this is complex 2nd, and this is the complex 4th. So as we have discussed, complex 1 is the NADH dehydrogenase enzyme complex. Complex second is the succinate dehydrogenase. Complex third is cytochrome BC1 and complex 4 is the cytochrome C oxidase enzyme complex. Here, as we can see that the molecule NADH is being converted into NAD plus, and here two electrons are being transferred to the FMN, that is the flavin mononucleotide, and it's get converted into the FMN. The molecule transfer from FMN to the ubiquinone molecule, and ubiquinone molecule again get reduced as UQH2 is shown here, and then this ubiquinone or reduced ubiquinone molecule transfer the electron to the complex 3, and here from here also the succinate molecule is being converted into fumarate, and the one electron is transferred from here to the complex third. And now complex third molecule, it gives out 4 proton to the outside or to the intermembrane space, and here from complex 1 also 4 proton molecule are being also ejected out in this process. Now the electron moves from the complex third to the complex 4, and here the final electron acceptor is oxygen molecule, and which get reduced into the water molecule here by the use of the complex 4 or cytochrome C oxidase enzyme, and here in this case, in this particular complex, only 2 protons are being ejected out. So the point to remember is complex 1 and complex 3 eject out 4 hydrogen or 4 protons each during the electron transport chain, while the complex 4 ejects only 2 proton molecule or 2 protons here. And so we can see that this is the complex 1, 2, 3 and 4. It comprises of the electron transport chain, and the last one that is shown here, this is the ATP synthase complex, which is responsible for the conversion of these accumulated protons into the ATP molecule. So due to the electrochemical gradient which has been generated here by the accumulation of these photon, leads to the conversion of ADP molecule into the ATP molecule. So in this way, the protons or the electron are being transported from complex 1 to complex 4, and finally the accumulated protons are being used for the formation of ATP molecule. And here in this particular diagram, you can see that the starting point of the cellular respiration was the glucose, which is being converted into the pyruvic acid by the process of glycolysis, and which then again leads to the Krebs cycle, and the link between the glycolysis and Krebs cycle was acetyl coenzyme A, and here the Krebs cycle again leads the chemical energy that is in the form of NADH and FADH2, and the next process, that is electron transport system, which has been carried out in the intermembrane space and the inner mitochondrial membrane, and gives out 36 ATP molecule. And here the oxygen molecule, that is the final electron acceptor in complex 4, is being converted into the water molecule. So this is how the whole process of cellular respiration is being completed in the process of electron transport chain. Now, what are the importance of the electron transport chain? First thing, it is the most important process of the cellular respiration, as it produces a large amount of ATP molecule that result in the creation of ATP molecule in the system, and the process is oxidative phosphorylation. Here, 4 protein complexes are being found, which couple the redox reaction and develop an electron chemical gradient. Okay, so it takes place in mitochondria in both the cellular respiration as well as in case of the photosynthesis also. The primary function of electron transport chain is to generate an electrochemical gradient, and the synthesis of ATP during and cellular respiration and in photosynthesis in mitochondria and chloroplast respectively. So here In this way, the whole process of electron transport chain is being summarized by us. And so thank you so much. If you like our video, please like and share and subscribe our channel for more such videos. Thank you.